Hi, my name's Sadie Ryan. Thank you very much for listening to my talk today. I'm going to talk a bit about work we've been doing looking at shifting risks, particularly about global change and vector-borne disease suitability models. And as you can see from my rather busy opening slide, this is work that I do as a collaboration with many people across multiple parts of my university and other universities. And just to give you a sort of flavor for where the framing for my work comes from, I operate on lots of different kinds of funding. And so some of these questions start out as entirely ecological theory and oftentimes end up as very directed applied questions in the sector of health. And so much of what I do is shaped by who's asking the question and what the goals are. And so that will become clear throughout my talk. In today's menu, I'd like to introduce you to a physiological model for malaria, and then I'll show you some maps of very straightforward things, and then we'll talk about some new old malaria maps, and then move into 80s transmitted disease models, and think about vector-borne diseases and climate change models and decision-making, and then think more about other approaches that we've taken to looking at vector-borne disease risk, and think about what that means we're mapping, and so what we're communicating. And then I'll have a couple of summary thoughts on utility and purpose. Importantly, I'm going to be addressing sort of the issues with thinking about how to translate models into maps and how our maps are talking to decision making. And so I'll keep sort of harking towards that. So scaling this all the way back to some biology, we started out about a decade ago asking this question of how does malaria respond to temperature with the eye of how climate change will impact malaria transmission. And so we start with what are the processes driving malaria transmission? Here we have the malaria transmission cycle. And what we can see is the mosquitoes sucking up the parasites from an infected human, those parasites developing in the mosquito and migrating to the salivary glands, ready to transmit to the next human they bite. And a lot of that process is happening in the mosquitoes. So the parasite and the mosquito together, and they are happening in the environment. And so they are very, very susceptible to environmental temperatures. And so temperature drives a lot of what happens with the processes that drive malaria transmission. And so when we look at the equation on the left, this is an equation for transmission made by MacDonald and then Ross MacDonald in the 50s. We see that it's constructed out of different rates. And these rates are the biological processes of the mosquito and the parasite. And a lot of these happen in the environment. So things like the density of mosquitoes, the biting rate, the infectiousness of infected mosquitoes, vector survival. And so what we can do is talk about how these processes are driven by temperature in the environment. Essentially, we are bags of chemicals, so are mosquitoes, and those chemicals make enzymes, enzymes make proteins. And so all of these rates are essentially the result of these chemical processes. So these chemical reactions, as many chemical reactions do, speed up with temperature. And so if we put this on a performance curve, thinking about how those rates, those essential life history rates, those development rates of the pathogen in the mosquito happen with temperature, we see that there's a minimum temperature before which nothing happens. Things are just not going. And then it sort of speeds up towards this optimum temperature, this TO, our optimal temperature. And pretty fast after that optimum, it all crashes as we hit this maximum temperature. And that gives rise to this classic curve-shaped response to temperature. And so what we did was we said, okay, these life history and pathogen development traits have been measured in laboratory experiments. We're going to go to the literature in this first case and locate those laboratory experiments. We want constant temperature experiments, recording those rates. And we're going to fit those physiological responses. We're going to fit those data with lines and curves. And we're going to compare those to see what the best fit is, because we expect that these are curve-shaped. And then we're going to take those responses and put them back in that R0 equation as a function of temperature. And then we want to validate that with field data. So a bunch of us went to the literature and we pulled out these rates from all these studies and we compiled them And it turned out that those best fit models for those data happen to be curves. In every case, they're not identical curves. As you can see, some of them have that right bump very obviously, some of them do not. But all those curvilinear responses to temperature were piled back into R naught. And so this is a function of transmission versus temperature. In every study I'm going to show you, we've scaled this to 1, so it's a function of the optimal temperature rather than a classic R naught telling you how many more cases are going to be generated by a single case. And what we found is that this new estimate that we had for malaria suitability of transmission temperature was about 6 degrees cooler than the previous estimate. And so to think about validating this curve, we wanted to have a parameter that scales with our transmission that is nothing that we've put in the model already. And so we looked at entomological inoculation rate in field studies, and we said, okay, for every single one of these studies, what is the month and location that this is taking place? And we pulled temperature data from a 40-year product and found the location and temperature in that month for that study and put those onto this graph. And so what you're seeing is the temperature at these study locations on our curves. And we found that that was a pretty good way to validate these curves. And we published the paper, and we mentioned things like climate change. But so what do we do with this information? How do we tell people about it? And it turns out people like maps. So if we're talking about temperature and temperature changes, we want to think about shifts. And so the question of where is really important for control, for intervention, for prevention, for surveillance. We might think about this as prevention maps for travelers. We might want to start thinking about anticipating shifts of transmissibility in seasons on landscapes so that we can do planning. And so we put this on a map. So I pulled down these global climate layers from WorldClim at the time. And these are basically telling you what the temperature is estimated to be in every pixel. And so these are roughly 10 kilometers squared at the equator size pixels. And I used an aridity mask, as in I took out where it's too dry for mosquitoes to even start their life cycles. And so that's grayed out on these maps. And I clipped this all to the continent of Africa. And so what we're looking at is in every month, monthly average temperatures translating into that optimal transmission. And so what we're seeing is this Goldilocks scale, as we call it, where the dark reds at the top are too hot for transmission and the deep blues at the bottom are too cold for transmission. And somewhere in the middle, it's just about right. And so as we march around these months of the year, we sort of see that this captures the broad geography of where malaria suitability, so the transmission suitability risk of that temperature, exists. In terms of being able to convey what this risk actually looks like on a landscape, we need to think of it in numbers of people, or at least that seems to be a good way to communicate this. And so what we see here was our first one of these sort of density suitability maps, where we're taking population density, we're taking our future climate scenarios, and we're saying, where does this most optimal transmission take place on the landscape? And in terms of how many months of the year do we see that? So what degree of endemicity of malaria predicted? And where are those sort of density and malaria transmission hotspots? And so we see this kind of marches across the continent as we go through these future climate scenarios. Now, in this case, we were in 2015. We'd started this work years earlier. And so we were using these older climate models. And so we're talking about a HAD-CM3. I think it's an A1B scenario, so sort of a mid-range scenario. And so it's important to note as we think about sort of climate change modeling when we're talking about this, that the models that we think about in terms of projecting future climate change also change through time by virtue of different policies, different ways of framing how we're going to do mitigation. And therefore, there are these different ways of expressing climate change impacts through time. And so this map basically gave us our platform for actually communicating what we meant by these models. And so that was kind of nice. So this idea that some parts of Africa could become too hot for malaria was picked up for the first time. And so we felt like, hey, maybe we've got, a tool for communicating some of this risk. Some of that group that originally worked on the malaria curves went forward, and we grabbed some funding to carry on and look at different diseases. So we decided to go for dengue to start with. And dengue gave us a challenge that there were two vectors transmitting dengue, Aedes albopictus, the Asian tiger mosquito, and Aedes aegypti, which is the yellow fever mosquito. And at the time we started doing this, there turned out to be three arboviral diseases having epidemics and then pandemics. In the case of Zika, that was declared a pandemic. And so we were thinking about this in the context of these emerging and re-emerging arboviral diseases in the Americas. And so what we see here is when we did the same process of constructing the curve, Aedes albopictus transmits at a cooler temperature. Its optimal is cooler, its lower bound is cooler, its upper bound is cooler as well than Aedes aegypti, this yellow fever mosquito. So we have these two slightly different thermal responses in that transmission curve. And because we didn't yet have a lot of the laboratory information for chikungunya and Zika at this point, we were validating using human case data. And so this turned out to be its own model complication that was very interesting, and you can read all about it in the paper. But so we had these two new thermal curves, and the question then was, okay, so how does this look under climate change? And at the same time, a lot of folks were picking up on niche modeling and producing these global maps to think about, can we predict what's going on with Zika? Because that was the pandemic of the moment that people really wanted to know more about, and we didn't know that much about it at the time. And so environmental suitability maps were made for when there were Zika cases. People could make case-based niche maps. People could use these Aedes aegypti maps to inform what's going on. People can use dengue maps to inform what's going on. And so there were a lot of global niche maps appearing at the same time. One person put the same human case data for dengue and for Zika into the same model and ran the two models and discovered that they don't look the same on the landscape. And so that gave us our first hint that maybe dengue and Zika were not the same. So let's pin that thought. Around the same time, I had been contracted by USAID's President Malaria Initiative to think about our malaria models in their context of new climate models and thinking about shifting demography on the landscape in the future. And so this slide captures rather a lot of this question of how to communicate risk using our models. As you can see, we have the USAID colors. Part of thinking about risk for malaria is understanding how people decide what is seasonal versus endemic malaria. And this matters for thinking about how many months of the year we want to describe that transmission suitability with these models. And what kinds of decisions people make in response to how we describe the seasonality of the disease. And so we used their framing for how to describe seasonal and endemic malaria, so how many months of the year at risk that would be. And this slide kind of captures one of the concepts that's complicated in a lot of this work, which is thinking about how we overlay our suitability models for risk onto demography to talk about people at risk, that PAR, people at risk. Because if we look at this graph on the left in our beautiful USAID colors, we tend to visually cue in on the red, and we see there's a lot of red in Central Africa. When we look at the graph on the right, what we see is that this question of the distribution of population density across the continent shows us a different vision of that risk, where we see Western Africa, where we haven't cued in with our eyes on that, in fact has far greater risk than Central Africa by virtue of population density. Now in this work, we also had some different questions to answer because there were new climate change models being considered. They were considering two representative concentration pathways in their decision-making framing, and this was a regionalized decision-making, and so we had to cut our maps up. And so that's sort of thinking about how we translate this work into risk a little bit more. And so we also consulted these things called SSPs, so these socioeconomic pathways. So what that is there's a really nice framing for thinking about these futures of where the population distribution is going to occur in response to climate change itself. And so as we're pushing our climate change models into the future, we're also pushing our population distributions into the future. So there's these theories about future resource distributions, potential conflict over those resources, and which kinds of mitigation pathways are going to lead to different socioeconomic outcomes, which result in different population distributions. And so as we start combining these models, we're really thinking about sort of what does the future really look like on the landscape, and how does that population at risk change? And so we're sort of thinking about these policy frameworks. I'm going to skip back now to some of this 80s work that we did. So when we first did our dengue curves, we said, okay, we've got two vectors. We want to look at these sort of high probabilities of transmission. For how many months of the year are these areas susceptible to that? Where is it suitable? And we see that our cooler mosquito, the Aedes albopictus, we see that that distribution of suitability kind of breaks up in South America, whereas Aedes aegypti has that nice all year round, bright red, 12 months of the year suitability for temperature-driven transmission. So what happens when we put that on our future climate scenarios? We see that under different representative concentration pathways, in this slide, we have 2.6 and 8.5, 2.6 being the most optimistic future, and 8.5 being our least optimistic, now called business as usual, pathway. We see on the left, we have Aedes aegypti, which is that hotter yellow fever mosquito, and Aedes albopictus on the right, which is the cooler mosquito. In both of these future scenarios, we see that northward expansion, the sort of poleward expansion that occurs with distributions, but only for a few months of the year. And we see a sort of breaking up of Aedes albopictus in those areas where we expect it to get too hot for transmission for albopictus. And so what does this mean when we translate it into the people subject to this risk or this suitability? When we put this onto thinking about people at risk, this is kind of a busy set of charts, but on the left, what we have is any transmission risk, so 1 or more months of the year. Aedes aegypti on the leftmost side, and Aedes albopictus on the slightly right side. And we're looking at 2050 and 2080 in the top and bottom panels of these. And this is looking at those different representative concentration pathways going along the bottom. So our most optimistic to our least optimistic, and across several different climate models. So we took different climate centers produce these different global circulation models as responses to those mitigation pathways proposed. And so what we see is we've got these increases in the billions of people over time for 1 or more months. And then if we look at this year-round transmission, that endemic suitability risk, we see that things get too hot for albopictus in many ways. And so we have this question now posed to us, which is, what does this mean for climate mitigation? We see that things will get worse in terms of Aedes aegypti transmission, all things being equal. But for Aedes albopictus, we see a decline in that suitability as it relates to where people live in terms of population density. And so we wanted to think about that more in sort of actual place-based context. And so we use the global burdens of disease regions approach. And so these are regions where health conditions are described. And it basically gives us a framing for thinking about, okay, can we say, where are the places that we will see the most increases in this example, year-round transmission risk under the worst case scenario? And so we can kind of see these different regions pop to the top of our priority list or our risk list. And so we see with this global burdens of disease regions approach, we can see which of the regions pop to the top as the highest risk under these scenarios. And we can discuss things like how many people will be exposed for the first time under these scenarios, and how many people are going to be sort of changing in risk in these regions. And Interestingly, we found that this is a really nice tool for discussing this with journalists, with people trying to, sort of use the implications of this to describe what the findings of the study are. But in a published paper, this ends up in a supplemental table because it's not that compelling a figure. Um, and so this is something we wanted to sort of work more on is, how do we communicate this kind of risk, which is much easier to glom onto in terms of figures. So I mentioned to hold on to that thought about dengue and Zika being differently. So as we were working on this, colleagues went into the lab and they infected Aedes aegypti mosquitoes with dengue and Zika in parallel and recreated those thermal response curves for the infection uh, characteristics as a function of temperature. And so when they first presented this, they said, look, the optimum is similar, and therefore we could be optimistic that our dengue maps work for Zika. However, if you look at the lower end of this curve, I thought, oh, I'd like to see this on a map because something's different. The Zika, which is that dark blue curve, is basically it has to be warmer for it to transmit, and that's going to mean something on a landscape scale in terms of the different risk from dengue and Zika. And so, taking what we knew from this paper about Zika transmission temperatures and thinking about what we had sort of distilled in terms of how we wanted to convey our risk concepts for future disease risk, we went back to our rainbow maps to say how many months of the year are these areas suitable for transmission based on that Zika paper instead of the dengue paper. And we see that the sort of the clumpiness looks a little different on the landscape, but that doesn't really tell you a heck of a lot. Using a 2015 population grid of the globe, there's about 5 billion people exposed to one or more months of risk under this suitability model. And then when we look at this in, when we look at this in 2050, when we're looking at this moderate and worst case scenario, so we're taking that representative concentration pathway 4.5 and 8.5 in a sort of midline global circulation model, what we see is these sort of poleward shifts happen. They do not happen as dramatically as they did for dengue. However, we still don't really have a way of visually sort of glomming onto this usefully in terms of risk. Why did we pick 2050? We picked 2050 because right now what we're seeing is that populations appear to have a sort of immunity to Zika. There seems to be some antibody enhancing immunity going on. And in 2050, we will be an entire generation removed from that. And so if something like Zika were to show up new, in this case, we're modeling it as Zika, we would be facing a naive population again. Recent work has shown that the Zika that showed up for us was a specific strain of Zika, and that maybe there are specific strain and mosquito matches that we need to consider more. And it's really exciting to see this work coming out of lab groups because we can think about how to refine the models to more appropriately talk about those specific pairings of the pathogen and the bug. But so what we wanted to do was figure out how to get that global burdens of disease risk analysis into a figure, essentially. And so in this case, we've used those global burdens of disease regions again to think about where are these people newly at risk in 2050 by region. And so what we see is that with mitigation, high-income North America experiences the greatest increase in new exposures, but without mitigation, Eastern Sub-Saharan Africa turns out to be the region that experiences the greatest increase in new exposures. So this tells us that there's going to be differential impacts with and without mitigation in terms of that distribution across the globe of where suitability will arise. So now I'm switching gears. All the work so far that I've shown you has been thinking about one axis of sensitivity to the environment, and that is temperature. Whereas we've masked out areas that are too arid, or we think about the risk to people from our Aedes mosquitoes in terms of where people are, because that's where the containers are that the Aedes aegypti mosquitoes breed in. We may want to think about this sort of multi-axis approach to thinking about the environment. And so species distribution models, or ecological niche models, those terms are used relatively interchangeably now. And essentially, this relies on the concept of niche. So, where you are has multiple layers of climate and environment, vegetation, soil conditions, all these sorts of things. And those layers of environmental data that we can measure comprise the place where you are, that you get your resources, your fitness. That is the niche. And so some of these models do better with a fundamental niche concept, some of them do better with a realized niche concept. But it is this concept of niche, of the different components and axes of your environment that give rise to your ability to be in that place, that underlies the principle of these models. So we use the known locations of occurrence and the underlying environmental conditions to predict geographic distributions in areas that are not represented by those occurrence data. And so in the context of public health and disease risk, this is often used to estimate geographic ranges and risk of exposure to pathogens, vectors, etc. And so for environmentally driven diseases, this is a framework that makes intuitive sense. And I'll get into a little bit of that. So if we then are able to use our climate predictions to think about future climates within the context of this environmental suitability, it allows us to extrapolate these environmental niche models to different time points so that we can think about shifts in exposures and risk. There are lots of algorithms out there at the moment for using ecological niche models. I'm going to show you a comparison of just a couple of them. But essentially how we do this in terms of workflows, you can take georeferenced species records, so that could be museum collections, public databases, scientific literature, public health surveillance, and field sampling. You accumulate your layers of environmental data, and you make sure that your grids are all in the same resolution. And then you use your algorithm to describe those conditions to project them to places beyond the sampled locations. And there are many methods for validation and for sorting out which models are most appropriate. But these are used widely in the literature at the moment. And so what we did to explore some of the questions that came up with, how useful are these for asking some of these questions, is we re-examined a data set that had been published as a Maxent ecological niche model previously, looking at Dermacentor variabilis ticks and Rickettsia montanensis pathogen in those ticks. And so what we have is both the presence of the vectors and the presence of the infected vectors as part of our occurrence data set, which is nice. And so to ask this question of, if we use the vectors, so the ticks themselves, as a proxy for risk on the landscape by creating one of these niche models, is the infected niche the same as the overall vector niche? So is your risk of contracting the disease the same if you encounter the vector on the landscape as if you encounter an infected vector? Are those niches actually different? And so if we think about things like fitness consequences of being infected, or what kinds of hosts that are the reservoir for the pathogen in the first place, where they might be on the landscape, we can think about the fact that maybe the infected vector's niche is not identical to the overall vector niche. And then this other question of, if we use the same data inputs and the same environmental layer inputs, are the models producing the same answers? And as you can see, the answers to both of these are no. So this is just a quick demonstration of the outcomes of using 5 different model types on the same data. And the yellow represents the ticks with R. montanensis, so that's the infected niche, as it were. Red is all of the sampled ticks, and then orange is the overlap in those distributions, so where the infected niche is in the overall niche. And so we can see that these 5 different algorithm approaches gave us somewhat different answers, and that the infected niche is a subset of that larger niche, but not entirely sort of in an elegant donut shape that we might expect. And so this is just breaking that out with sort of all of the ticks and the positive ticks and their probability of occurrence in this gradation map. And so we need to think about sort of how will people translate this to risk? And right now we're looking at ticks as a sort of, a newly a newly highlighted risk on the US landscape, at least, and thinking about tick-borne diseases. And so when we think about the tools people are using to think about risk, we need to know sort of how that translates into decision making and what kinds of things are actually being mapped. Um, and so I'm happy to chat more about that in the workshop. I wanted to give a few sort of round up thoughts on the utility of mapping approaches that I've highlighted here. Um, One thing we've encountered is this question of the scales of maps and the decisions that people can make don't always align. And this is really important in terms of vector-borne disease management. Um, on the right, you can see these 6 little panels. And what this is when Zika was affecting the Caribbean, we were asked to review global Zika maps and say, sort of what they show for these 2 islands. And so these are just three of the big published maps. And as you can see, that's not a great tool for decision making. The maps are basically saying it's either mostly suitable or entirely suitable for Zika, and they really knew that by then. The other point to make is that there are never enough data when we're trying to make calibrated models using this data, particularly things like these thermal experiments that require a lot of resources and knowledge about how to do things like raise lab colonies of mosquitoes and infect them and demonstrate transmission. Data are also massive when we're thinking about things like environmental layers for the entire globe, and they're messy. And so, one message I have is be kind to data managers. Oftentimes, things like health data are very, very hard to use and require a lot of careful consideration and processing. So different data managers are encountering different things, so be kind to them. And understanding the scale at which biology matters really matters. And so with our tick models, air temperature isn't really what's going to be driving these ticks necessarily, although air temperature is a proxy for that environmental temperature, but ticks are really in the ground compared to mosquitoes flying in the air. And so we discovered that, incorporating things like soil layers is going to be much more informative when we're thinking about these ecological niche models. And when we think about sort of what scale at which intervention takes place in terms of vector-borne diseases and prevention, the biology of what is going on in the environment is really important. And I would even add that the behavior of people in the environment is going to be really important. One thing we discovered is that this ecophysiological maps approach using this temperature-driven vector-borne disease approach works across many systems. On the far left, you can see some maps of Anopheles stevensii using the rainbow months of the year way to talk about this. And if you look to the far left of those maps, you will see the Horn of Africa. And currently, a huge concern is Anopheles stevensii, these Asian Anopheles mosquitoes showing up in Ethiopia and other parts of Africa to transmit malaria because they are happy urban Anopheles. They breed a bit more like Aedes aegypti do. And so it's an interesting new invasion of a vector. In the middle here, we have a map of citrus greening disease, which is another vector-borne disease. And this, speaks to economics of citrus groves. And so we can think about anticipating where this disease might be more suitably transmitted in the future, where we might want to get out there with new technologies for treating it, which are coming up. On the bottom here, we have a map for blue tongue in sheep. This is a midge-transmitted disease, and we're thinking about sheep on the landscape. And so this is FAO data with sheep densities instead of human population densities. This is great because this is one that actually does have vaccine preventable interventions. So thank you very much for looking at my talk. I hope that the models and tools that I've presented here provide some really good conversations for the workshop. I'm really excited to meet everyone else and think about how our tools work together. And with that, I will say thank you to all my co-authors and collaborators who have worked with me over the years on all this work.